doubling up on some of the bio boxes, we do.
Is there any slurp capabilities? It looks like it probably will scrape off the rock.
It looks all full.
Yeah, I don't think we're not supposed to mix the sponges, but we could mix sponge with coral, from what I understood.
Roger. Yeah, this might be, if I scrape it off the rock with the slurp, it might preserve its integrity. What's in the forward boxes? Are those both sponges?
I think they're all
like
full with sponges.
They're all sponges, it looks like.
Roger, so you can't really do a slurp and drop.
Yeah.
Yeah. I don't think we're supposed to mix because of the spicule cross-contamination and things like that.
Yes.
We can try scraping it off the rock. And is there any coral in the starboard bio?
It looks like it's all sponge in there.
It's just, I think every compartment is sponge, I think, is my understanding.
Yeah,
the
that's what
flush
I
has
understood
sponge
as
in
well. Yeah.
All right. Well,
Can we use an Iskin bottle?
Does ROV have an extra sock you could put it in?
Can we do a push core?
Oh, Abby just popped on.
To it?
Sorry?
Hello, Abigail.
Oh, sorry.
Abby, what is that? So the specimen that we're looking at right now, what would you say that is?
I don't know if she's on mute or.
She took a nice screenshot, she said. So for those who are listening in, we are chatting with Abigail via Science Chat, and that's just all text. It's texting back and forth. Okay.
I don't know if it's, even if you can't sample it, guys, do you think it's worth just grabbing it and getting a closer shot or moving closer?
Yeah, that's a good idea, Chad.
What did you say, Chad? Sorry, I was noticing.
Yeah, I mean, so, Marley, you want to come wide there, please? So,
I mean,
you can grab and scrape it off the rock if you want, but,
It's 5 centimeters.
We have both our Wonder Sponge experts now in the Science Chat, Abby and Amanda, so.
Yeah,
we're
going to
gonna
get a little closer, I think. Pop up a little bit.
Be able to get their input.
Yeah, I think at least just getting a little closer and getting a tighter shot would be better than nothing.
Roger that.
They're in complete support of that tighter shot.
I think they're all full of sponges.
Hi, Amanda.
So she's calling it a semi-infaunal sponge, which is a term I've never heard before.
So somewhat in the sediment.
There is a slurp with a coral in it if you want to see if it could go into that jar. Up to to leads, though. We have a slurp number 4, only has Paragorgia in it, I think. Ooh, is that what? Yeah, that's what it says.
All right, Marley, full on in there.
Abby's saying it. Yeah, Abby's saying it could go in there.
Great.
Let me just double check that it's Paragorgia.
Okay.
I'm going to check the data recovery file too.
All right. It would be sample 73.
Okay.
Yeah, my sample sheet's saying Paragorgia.
Yeah, mine says same thing.
Got any more in there,
Paragorgia.
Marley?
Yeah, that's it.
Reg.
Then I think we should be okay.
Oh, wow. This is so cool to look at. So different than anything I've ever seen before.
It's almost like a bunch of little eyeballs or something.
So, Mary, did you check that out for us? Was that you, Mary?
Yeah, so it looks like if
thank you.
You want to put it in a slurp, we could pair it with slurp 4 because that only has a Paragorgia in it. Roger that.
Roger that.
All right, slurp number 4. Marley, come wide, please. Awesome.
So this would be sample 084.
Hello.
just going to do it
Yes,
really
084.
Whoa!
Thank
Oh.
you.
That's an amazing shot.
It's always
wow.
nice
That's
with
beautiful.
the black background, yeah.
Went right to us.
Oh, wow.
Don't come too close, buddy.
Oh.
Don't go. No.
Where'd he go? Are you inside of us right now on the porch?
Yeah. Oh, he's on the porch.
Back it up.
What?
Back it up.
That's
Yeah,
an interesting
he's on the
decision.
Back it up.
He's backing out.
Aw.
Like that habitat.
Yeah, buddy, you gotta go.
Go.
There he goes.
There he goes. He figured it out.
There we go.
man.
Oh, poor
Oh.
thing.
Alright, let's just
I
not
know
look.
if he goes.
Poor
Oh, no,
buddy.
he
Oh,
seems
that's a
very
female.
upset.
Poor
That's a
guy.
female. All right.
Alright.
Poor
So sorry,
gal.
that's 08. What was the sample number?
084.
804. Thank you.
we said
First
guy.
hit.
I said it's a gal, but Kurt made sure we got that right. It's a she. That's right.
Poor little girl.
It's a female skate. That's right.
So we're going to do a scrape and slurp? Is that what we're doing?
Oh yeah.
Yeah, one thing Amanda was pointing out is to try and, when we zoom in on it, kind of just touch it at first, just to see what the behavior is, if the oscula will retract or move.
Roger that.
And is this a demo sponge? Is that what we think it is?
There was some discussion. Of
I went
that.
back in the, yes, Abigail said yes. Okay, thanks, Abby.
I think it's pretty interesting. Most people probably don't think sponges have behaviors, but remember they are animals, not plants, and they can respond to touch. Some of them, anyway.
And Mary, which jar is it going into?
It's going to
Or
be,
is it going into a box?
it would be jar 4.
Okay. Thanks. And I'll make note that it's in with the Paragorgia.
So jealous you guys are going to get to see it in person in a little while.
So she says, poke it while they collect it, but a poke with some zoom would be nice. But I guess that's what we just went over. Okay.
All right, Kim, you want to go ahead and flush? And stand by for.
Hush.
Mary, I'm assuming some of your stills already had the lasers in them. Is that right?
I'm sorry, what was that? You kind of broke up.
Oh, sorry. Do some of your still images of the sponge have lasers in them already?
They should. I can't quite look back. Let me see if I can look back on those photos.
Thank you.
All right, Ben, you want to press stick lock? Thank you.
Oh, so they're saying this is the moldy cheese sponge.
Yeah.
It looks very different than the other ones we saw, to me.
All right. And sample
Says
jar
it's hard
number 4,
to sample.
correct?
Yes, Jess.
Roger that.
Yeah.
Mm.
Nice. Getting down
4,
6.
7.
Yeah.
Flush.
Number
1,
4. Yeah.
2, 3, 4.
Nice. All right. Be a little bit of a bad, weird grab, so.
So we're going to zoom in and poke it and then
Yeah.
try and scrape it after that? Okay.
Actually, if you guys don't mind, I'm going to reset my jaws on this because it'll make it hard to see when we poke it.
Okay.
That's all good with you. Apologies.
Yes. No
So,
problem.
or maybe we could first poke it without the slurp.
Oh, sure.
There's
Yeah, we can do that.
something, and then go back, right?
Sure thing.
If that,
Sure.
we have some time.
Stand by, Marley. Stand by.
Oh, sorry, I thought you said go.
No worries.
Thing is tiny.
Yeah.
I know.
All right, Marley, go ahead and see if we can get an image of that. Roger.
Maybe it's like an iceberg and all of it's underneath. She said semi-infaunal.
But then I asked her why it was hard to sample, and she said it was very thin.
Well, you see, the oscilla
we'll
looked like really flared and skinny. Interesting.
One quick.
There we go. All right. That is all the way.
Roger.
Okay.
Knock, knock.
Does it scream?
Yeah.
Change. All right, one little toe tap.
This is very delicate work.
Mm-hmm.
Sorry, let me kill a little of my vertical thrusters. Yeah. Just killing a little of my verticals. Oh, it's contracting.
Oh, yeah.
Oh, no, that's just me.
Wow.
Oh, yeah. Oh, yeah.
Oh. You're depressing it.
Pretty spongy.
Indeed.
Go figure.
I don't think it's a coral, guys.
You sure about that,
We're not
Marley?
in coral land anymore.
I think that was probably enough, right?
Okay. Roger that.
She asked, she says that was pretty fast for a deep sponge where it's so cold for it to contract that quickly.
You want me to press it one more time to see how it contracts?
She asked if we could retreat and watch for 20 seconds or so.
Roger.
I think she wants to see if it
Comes back.
changes back.
Interesting. She knows all the tricks.
This is input from Amanda about poke and then retreat. And watch.
Mind looking over at the arm there, Kim?
Oh, so she said that's the...
Yeah.
Go ahead, Lisa.
Jen. Well, I'm just reading what Amanda wrote. The oscula farther from the poke are closing now. That is what I am
everything's
watching.
closing up.
Awesome.
Okay.
Yeah. This is like behavioral work, deep sea behavioral research. And look at all the people working on this together.
How many people does it take
See
to
how
poke a
this-
sponge?
I know. Exactly.
Well, when it's a deep
And
sea sponge, a lot.
That's true.
Yes.
I mean, that was impressive.
What depth are we now?
We are at 383 meter step.
All right, I think we're good to collect.
Roger that. Marley, you want to come wide there, please?
Yeah, do you want full wide? What are we doing?
Yes, ma'am.
Can we zoom in with the lasers on while we're doing this?
Lasers might
Or
not
whatever's
make it.
best.
We can try.
Whatever's best for your operations.
Oh.
All
right, that was interesting.
What happened?
Not sure. I killed hydraulics to the arm, though. Interesting. All right.
Yeah.
Let's try that again. Maybe Kim, if you want to be on the kill switch.
That wasn't the grip. It felt like.
No, not sure what that was.
Yeah.
All right.
So that movement happened independent of you?
Yep.
Yeah, it looked like it had a life of its own.
Robots these days.
Oh.
The thought of them moving on with a mind of their own could keep me up at night. So.
Oh yeah.
There's a whole.
There are movies about that.
Based on that. Yeah. I'll be back.
And if anyone's wondering why we're, how we decide what sponges to collect, well, you've probably been hearing that there's some experts who are working with us who do surveys up and down the coast and are looking at all these different kinds of sponges and trying to identify them. And so there's photos of this sponge. There's been one possible, I guess at least one has been collected, but sometimes it takes more than one to help figure out what kind of sponge it is and what it's related to in the exact species and describe it. So
That's a good point, yeah.
We better understand
while you do that,
what's out there.
I'll do a zoom on this guy.
Thank you.
Marley, you want to push in on there on the sponge? We can get the lasers kind of on it.
That's the flash.
Now it has a completely different shape.
Come a little
it's
way.
not as round and pink cushiony. It's more flat.
Roger. Roger that. Awesome. Roger. Roger that. Riley, gonna come a little wide there. Just want to see how my hand's oriented. All right. All right, go ahead and push it in there a bit, Marley. That's great. All right, Kim, I'm ready for you. Roger that. Ooh.
Can we see the slip jars on 3, perhaps?
I'm not sure we have.
Yep. Nice. Let's see if it gets in.
You got it?
She got it.
It's not in the jar yet,
Yay.
Oh, yeah.
but
There it is.
there it is.
You can see it?
Oh,
Yeah.
yeah, awesome.
Blender shut off, Roger. Go ahead and come wide, Marley.
Where did that go, though?
Awesome. That was a cool sample.
Nice job, Jess.
It's very small.
I know.
Nice.
Well done
Yes.
with the robot.
And so interesting. I'm kind of bummed that Death Star sponge didn't stick, but I guess we'll go with moldy cheese.
Moldy cheese. Is that the moldy cheese?
Oh, was that the moldy cheese?
Yeah.
Yeah.
Oh, got it, guys.
Yeah.
Yeah. I'm just as surprised as you are, Ben. I was looking for something totally different.
Yeah, it didn't look very moldy to me.
Would you eat cheese that looks like that?
No.
No,
Okay.
but we were thinking of like the good yummy mold. Like I was thinking
Oh.
or blue cheese,
Blue
or that's
cheese,
what I was
yeah.
It
thinking
was
of.
like a gray
Veins.
blue tinge it had.
Yeah.
That's it.
Not that. That looks like an eraser color, you know, gray and like silly putty.
Yeah, I found it interesting for other reasons, not for its mold.
Yes.
Yeah, so they're commenting in the science chat the last time they tried to get the sponge, they had to grab it instead of slurp it, and it was not nearly as complete or of a sample. So this is, I think, the sample has caused a lot of excitement.
Good.
And great job.
Yeah.
Great job.
Ah.
And Lisa, just a time check. I
Yeah.
think, guys, correct me if I'm wrong, Jess,
6:43.
poll time's a little over 30 minutes from now.
Correct. That is
Mhm.
correct.
Yes. Thanks, Chad, for the reminder.
Do you guys
So
want to look
would-
for some brachiopods on this rock?
Yeah.
I think we
I don't think we're
looked,
gonna have-
and yeah. I don't think they're here.
Just
At least-
yeah. And I don't think so, Chad. Now that you chimed in, there's probably not time to do a photo mosaic, even if we found a likely candidate.
Correct,
so
unless we had
should-
it right now. Yeah.
Yeah.
Or a very
So maybe
small
we
target.
should just. Yeah. Maybe we should just try and find some brachiopods then. So if Daniel is still on, or we know what we're looking for, maybe we could do that.
Sure.
I'm looking at we need to do a Niskin on Ascent. We did 2 transects, that's great.
Marlon, push in.
We're
please.
not likely going to get the photo mosaic, but maybe. And if we find shell hash or any brachiopods.
Roger.
Yeah, and I mean, we did mention push cores, but my assumption is this sediment is not deep enough for that.
Yeah.
Yeah,
Those
it might
rock
be
Thanks for
somewhat shallow.
It's probably just yeah. And it's pretty loose, and so it probably wouldn't stay in the in the sediment core.
Yeah. So I guess if we don't find the brachiopods, we could still do a scoop. And I think we did a scoop on the earlier shift, 4 a.m. to 8 a.m., didn't we? Yeah. So if we find shell hash, we could do that too.
So should we
Okay.
continue along this ridge?
Yeah.
Okay.
Yes.
Sure thing.
I'm going to do 305.
305.
Is that all right?
Roger. Yeah, Marley, you want to come wide?
Okay. I mentioned a little something. I don't know if Lisa and I would be able to participate in this, but as you get going, Amanda said on the ascent, if you can see that sponge in the slurp jar, it may expand back to its full size.
Oh.
She said they found that demo sponges with active behaviors were very dynamic and often inflate their oscula during ascent.
Interesting.
So.
Help me.
Interesting.
30.
Oh, Daniel's saying a scoop here would be fine.
So, okay.
So maybe at the next place that we look? Or should we set back down?
Yeah, so I have a question for you guys. So the scoop has a mesh bag on it, and if we do it in 2 spots, I would assume that would contaminate the first sample and make it invalid. Is that correct?
I'm not sure that he needs them to be differentiated that way. I think he's just looking for shells, but I'm not as familiar. I can look at the, hopefully he'll chime in here, but I'll also look at that sampling.
Shells independent of space.
And when I said that we had done a shell hash collection previously, it was on the morning shift.
Yeah.
With the scoop.
So if the scoop was used on the morning shift, then it probably has a bunch of samples already in it. So I would be curious if they would want to use it twice.
I don't have a sample sheet that says we use the scoop.
Maybe that was the previous dive.
We did the last dive.
Oh, maybe it was the previous dive. Okay, yeah, great. Thanks. It's all
Mixed sample is fine as long as it is from plus or minus 100 meters in the same area. Well, so the sample that Jess is referring to was taken a long time ago. Was that on this dive or did you confirm that?
I think they're right. It was yesterday.
Okay, it was on the first dive?
On a different dive when we were on, not Poti Peninsula, but Anacapa.
Oh, okay. Okay, so then we're good. Okay.
Yeah.
All right.
That's a rose thorn, I believe. If any fish people are out there, it has the coloration of a rose thorn. Rose thorn rockfish, very pretty.
Maybe we can try and find another ledge to look for brachiopods and then scoop some sediment from under that.
Ooh, hagfish.
This red bit here. Were those brachiopods in those images, weren't they kind of a brownish red color?
Yes. Sort of the size and shape of a clam.
Riley, you want to push in there a little bit, please? Where the lasers are. No, I don't know what that is, but it's not a brachiopod.
It's a brittle stars. Oh, and Daniel Geiger is saying that they are some are white and some are reddish.
Roger. Roger that. Marley, you want to come a little wide there? We'll look for some more brachiopods. Also, Marley, I think the wire cam went out again on the after the ship.
Lovely. Thank you for the heads up.
Passing by some great examples of squat lobster. They always look like they're just sort of that they've worked out too hard and they're just resting with their arms in front of them. It's very heavy.
So these brachiopods would be more located underneath rocks? Or would they?
Yeah.
Okay. Marley, you want to come full wide then, please? Thank you. That's cool.
Oh, yeah.
It's a very interesting habitat. There's just so much to see in a wide view, but then when you also, you know, do a zoom in, that you just see so much.
Super
Yeah,
fun,
and then you can even back out and look through at Argus and get an even different perspective.
Yeah.
Yeah. We definitely haven't run into, you know, big corals, but up here. I guess we haven't gotten to the tippy top yet. Do you want to run up to the tippy top, Lisa?
That sounds good.
Before we run out of time?
Yeah. I think let's just try and see if we're here, might as well try and see the ridge before we need to depart, which will happen in 25 minutes.
Yeah, it's just the tippy top of this ridge. It's probably not a steep slope. I think the steepest slope might be below us, but not here. Closer to that AD4 target. Let me check. Yeah. Yeah,
Thanks
we'll
for
be able to
checking.
keep to the side of this local maximum. Looks like one side is a little slopey, the western side.
Okay. I see that
good.
on my
Slope
path.
map.
Thank you.
Yeah.
Yeah, the slope map.
Great.
Is that what that background was you were just
Oh yeah,
looking at?
I can leave it up too.
So the purple is a more steep slope?
Purple is flat, so
Oh.
blue,
brighter
light
colors.
blue. Yeah, light blue is the best. So that ridge we were going along just had light blue. That was the slope. But again, this is an average over a good sized area, so the local features might be more interesting and fine scale. But
Well,
it's a good
that's a
indication.
Paragorgia with the polyps out that we just passed.
Yeah, isn't that nice?
Yeah, very pretty, very different.
going to change this just to 300. That's all right?
Sure. Get us that more slopey area.
Sea cucumber, little pinkish, tubular animal passing below. I haven't seen that many of that type of sea cucumber.
That's the first one I've seen.
You guys saw one or two
295,
before
Rod.
now, but you do often see them in
Actually, you want to
more
hold off that
uniformly
ship move?
soft
No,
bottom.
there's a big rock right here, potentially.
This might be the slope.
Might be the slope. Looks like an Argus.
Oh, you got an Argus hit? Oh, yeah.
No, just a slope. Never mind.
These flatfish are doing a very good job of blending in here.
Oh.
But we have one just about to pass out of view. Another sea cucumber and some rock covered in sponges
I don't see
and
any overhangs for
sea
brachiopods.
stars and brittle
I'm
stars.
sure it's what's on the other side of that, but let's just keep.
Okay, I'll come around to 295.
Do you have enough room to just poke over that for a sec? Or?
Oh, bat star. Those are the ones that look like a sheriff star. With his badge.
I haven't called in the 295, so.
Roger that. Like it
Yeah. It's neat.
flattens out.
This is very neat rock structure. Lots of complexity.
Yes, lots of places. Excuse me, lots of places to hide. If you need to hide. If you're tasty for other things to eat. In other places, you see the sponges are out filtering water, catching marine snow and plankton as it flows by.
Oop, that was a grenadier lifting off, I think. Very dark.
Didn't get a good look at it.
All right, I'm going to call in 295.
295, Raj.
Ooh, is that the mushroom coral or right there that we're passing?
Yeah, it is a mushroom coral. All the polyps are out. Have you not seen many of those on this
No,
dive?
I have not.
So that's the one that looks kind of like a golf ball. And out of the depressions of, or a wiffle ball, you've got soft polyps that are extending out.
Oops. Also, let us know if you guys see a rock that you want us to sit under and kind of look into for more brachiopods.
Okay.
Sorry?
Oh, I thought I saw something.
Yeah, looks like we have a couple little ridges in the sediment.
No, it was on the other side of a rock.
Yeah, and I guess if we set down, if we see some shell hash, maybe we should try and collect it now that we've confirmed that we haven't collected it before, so we won't be cross-contaminating.
Roger that.
So let's just have our eyes peeled for that.
Roger.
Thank you. We're going downslope, I imagine.
We're just going along the ridge here, so that we shouldn't be going downslope or.
Yeah, we're keeping a little.
Okay, it just seemed a little bit darker than usual, but.
Yeah. We did come off a little bit of a small little ridge, but it's only a little hop off.
Yeah, just little, not a lot.
Yeah, I'm hoping in about 100 meters, we're on more of a slope. So. A chance for some.
Oh, these sponges are really
Big.
impressive looking as little columns kind of telling us the way to go. Follow me.
Yeah.
Pop, and you want to stop
yep.
the ship until I come in front?
Sorry, I saw that.
Thank you.
So Lisa, I was looking at the protocols for the Santa Barbara Natural History Museum, or Museum of Natural History, and it says shell hash / grunge. What was the?
Yes.
Did you get any input on what the grunge component means?
I think it's another way of saying shell hash.
Okay.
That's the way I understood it.
Okay.
It's not a rock music from the Portland area.
Okay.
Yeah, that's the way I understood it.
Okay.
It's not Kurt Cobain's band.
See a brachypod?
Just see a white mass. I don't know.
On these larger rocks in front of us? Seems like they've got some ledges we could look at.
Yeah, I don't think that's a shell though. Just look at.
No.
Marley, you want to do a quick zoom there?
Sure thing.
Yeah, sponge.
Right.
Yeah, so Daniel Geiger is saying, so any sediment with biological components equals grunge equals shell hash.
Huh.
That seems like a lot of any sediment.
Well, so I'm asking if the sediment under this rock would be something that he would want then. Yes, perfect.
Yes. Okay.
So so target
Let's
straight ahead.
That fish. Yeah.
Right,
no,
Marley,
we're gonna gently request that the fish move ahead. Right? Hey, fishy. Let me get in there.
You don't know what's in store for you.
I want to move just a little onto the other side of that rock. But I think so.
So Jen, have you ever seen them do the scoop?
I did see the scoop happen. I guess it was yesterday. But I was in the middle of something else, so it was kind of out of the corner of my eye.
Yeah.
I have not used the scoop.
Yes,
before.
And I shared with everybody yesterday, I guess it was, that when I heard scoop, I thought like a scoop of sugar. It would be a spoon that came out and scooped it up. But it's quite a big bucket. So.
And Mary, is there anything that's floaty in the starboard bow box? Oh, Mary, I don't think you're on a spiel.
Sorry, I was muted. I think we're good.
Roger.
Floaty.
Roger that. I am floaty. That's what it is. Try that again.
So this is sample 85?
Rock under you.
Yes.
Yeah.
And we're putting it in BioBox.
Sit on marbles.
I don't know if I want to land on the others.
All right. Let's see if this works. Raj. Mary, any inclination as to what compartment the scoop is in? Is it an E or F?
I'm not sure.
Roger. Yeah, we can go ahead and pop it open if you'd like.
Remember yesterday I was in your starboard E, or the right it was?
It's not there. Ben, you want to keep an eye on the porch, please?
Yep.
Let me know if you see anything moving.
Roger.
So we just looked in the bio boxes. So
All right, we're moving.
Roger. I'm not seeing a scoop in there, guys. I'm seeing a sponge in F and no scoop in E. I don't think we have a scoop in this.
Oh.
What is that in E?
I don't know,
Yeah,
but our scoop has a little ball associated with it.
yeah.
And we would be able to see it in E.
it would float up.
The ball would float up, or else we would see the scoop when it's in the E compartment. I don't think we have it in this.
Okay.
Oh, well, there you go.
Interesting. Sorry, Daniel. You'll be able to grab it this time. I'm just looking.
We'll make sure that we have it for the next dive.
You didn't see it, right? That didn't
So
look like it was in there,
I'm looking at the dive. The dive plan said forward box standard, and then it said mesh scoop. So if it was going to be somewhere, I guess it's supposed to be in the forward box.
So usually we'll put it in the starboard bio, but we already looked in the forward box.
Okay.
Sorry about that.
I think so. I'm looking at the dive from yesterday. So it was put in box F.
Yeah, so F had a sponge in it.
F and E both have sponges in it right now.
It could have just been that it didn't get put in during pre-dives. We can pop it back open again and then share the camera view just to get a visual confirmation for you guys.
No, we're okay unless you need a visual confirmation from us that's better than your visual confirmation.
Let's just pop it back open and verify. That's great there, Kim. Yeah, so F has looks like a rock and a sponge in it. Is that correct, Mary?
Yes.
And E has some small sponge in it.
Also should have a rock and sponge in it.
So that would be that gray bit in there. Yeah, so our scoop's pretty big, so I don't see it in either E or F.
That's good. Yeah, you're right. It is a big, it's not like I said earlier, it's not a little teaspoon. Oh, it's like a cut off. To me, it looks like a cut off bleach bottle. So those are big.
We'll make sure to get on the next pre-dive. Sorry about that.
Yeah, it's no problem. No worries. We'll get it on the next dive.
Roger that.
So we have about 5 more minutes.
That is correct. 5 more minutes.
Why don't we, I don't think we don't have any time to collect anything in 5 minutes. So why don't we just try and get some great shots and enjoy the last part of Poti Peninsula, which has been a great place for us to survey and see an abundance and a huge variety of sponges and corals. So I'd like to stop on or end on a nice view of the Poti Peninsula.
Sounds good. Lisa, I'm just gonna restart my V-Link again. It got a little unstable, so I'll be right back.
Okay. There's a flatfish right in the center view. Do not know what kind, but it is a, oh, can't tell, left or right-eyed. I couldn't tell where the mouth was.
I would just go up slowly.
Roger.
I missed it. You're talking flatfish.
Yeah. I tried to use your pointers to look at where the mouth was, and I couldn't, it wasn't moving, so couldn't tell if it's moving, which way it would be going.
Okay. Odds are right-eyed. There's a lot fewer left-eyed flatfish.
Looks like we'll
There
have
are a couple
at least a rocky
species that can be right or left-eyed.
Oh, is there one?
This little rocky outcrop here.
It's like right on the crest of the bridge. We get some rock.
Oh yeah, if you're looking at the Argus view, it's moving up. You get a little wall. See? Ooh.
So Lisa, again, the Poti is named after what? Poti Peninsula?
So Matthew Poti, and I should look this up, he is doing some modeling, doing habitat modeling. In many ways, it didn't sound that different from what Ben was describing about his work, sort of using physical characteristics and data on the presence and absence of corals. And he has come up with a habitat suitability model to help predict where we would find deep sea corals and sponges. But different from your work, Ben, in that it's on a substrate.
Money shot.
Garden.
Kim has an awesome view for you guys in Argus.
It really is.
Oh yeah, that is very nice. Thank you.
Cool.
Dripping sponge. Like a dolly painting.
Overlooking Poti.
Yes.
And you want to start talking to the bridge and seeing what heading they want to cover on?
Yeah, we look like we're heading into the wind and the current right now, so just...
Oh, it's like we're pretty set up.
So folks who are listening, we're at the end of our dive here on the bottom of the Poti Peninsula, but there's still a part of the dive left as we ascend to the surface where we have the Nautilus ship. And when we ascend, we can see some very interesting pelagic organisms, so animals that live in the middle of the water column. And Ben, now that we know that what you work on, maybe you can narrate this part.
Oh man.
I got you there.
No pressure.
I'll be
No,
like,
you don't have to narrate.
no,
But it would be great if you see something that, please, just share your knowledge. That'd be
yeah, for sure.
All right, guys, it seems like that's the end of our dive, but at least we got some nice imagery at the very end there. Thanks to Kim. All right, so you want to repeat that bearing for me?
Yeah, so
Or heading?
we're 275. We're right into the current's like 0.3, went to 8.5 coming from 296.
Roger that.
So we're like
This is a
in it.
great, this is a great ending shot to pull away from. Wow, look at that.
Argus looks awesome too.
Yeah.
Oof.
Yeah, look at that diversity.
All right, so office.
Bye. This is really like great cinematography.
That's dramatic.
Yes, as we're pulling away.
Mm-hmm. It gives you a really nice perspective.
Yeah.
The number of sponges that basically we're just consistently seeing through this dive has really been phenomenal. I know we were also looking for corals, but I think the sponges did kind of steal the show today,
Yes.
as the bio boxes can reinforce.
Yeah, we had no room.
Stole the show and the space.
Yes.
All right. We might get a little bit more of a show as we stretch out. Very extensive.
So are we headed, like, what direction are we heading right now as we're pulling away? But we're
Yeah,
seeing
so Argus is
it
going,
still?
or sorry, Argus is facing 275 and Herc is going 095.
Okay.
East and west.
Yeah.
There it goes. So this is very different than our Davidson dives also, because we would have 3 hours of blue water ahead, and here we have 30 minutes.
Yeah.
It's just gonna fly by. I was actually in the past I've gotten a chance to work with Jess, and I was thinking about your work yesterday because a friend of mine's daughter is looking into going into a bioengineering major, and I was thinking in my head, who have I talked to about bioengineering?
Yeah.
And I was thinking of that gecko-inspired design. Really fascinating.
So are both of you at universities on the East Coast?
Bridge.
Stand by just a second, science. They are engaged in